Hello everyone, welcome to sqadia.com. I'm your presenter, Dr. Sannan Rehman, and today the topic of my presentation is muscular system. Now we'll be talking about the first section of this presentation, which is the skeletal muscles. This section is further subdivided into introduction, the origin and development, and innervation of the skeletal system. I hope you are excited about today's lecture. Let's get into it. So first of all, before getting into introduction, I'd like to refer to the figure given below. There are three types of muscles shown in this figure. The first striated muscles are the skeletal muscles. These are longitudinal fibers with a single nucleus within each cell. Then the spindle shape, the second figure is the spindle shaped muscles which have a single nucleus within each cell. These are the smooth muscles. Lastly, we have branching filaments with multiple nuclei within the filament. These are the cardiac muscles. Now, let's take a look on the introduction to the muscular system. So, the muscular system develops from the mesoderm. The skeletal muscles originate from paraxial mesoderm. The cardiac muscles develop from the visceral splanchnic mesoderm, which surrounds the heart tube. It's very important to remember the origins of these muscles as the clinical correlates would be related to them. So, the differentiation of smooth muscles or the development of smooth muscles is a bit different. It has three different origins from where it originates. First of all, it originates from the visceral splanchnic mesoderm surrounding the gut. Hence, all the gut muscles originate from this region. It also originates from its derivatives. And ectoderm also gives rise to a few smooth muscles. So, taking origin and development into a little detail, there are 7 somatomeres in the head which form the head musculature. Now, somites give rise to musculature of the axial skeleton, body wall, and limbs. You must remember what axial skeleton was. The skeleton was initially divided into two groups. The axial and the appendicular skeleton. The appendicular skeleton is the skeleton of the appendages. It also includes the shoulder and the hip girdle. However, the axial skeleton is the remaining skeleton, the skeleton of the central body that includes head, vertebral column, ribs, sternum, and vertebrae. So, they are arranged as somatomeres from occipital region to the tail bud. They undergo segmentation in which the caudal end of the upper somite and the cranial end of the subadjacent somite they use to fuse. After segmentation, they undergo epithelialization. A ball of cells with a small central cavity is now formed, which is called the somite. Now, the ventral region becomes mesenchymal to form the sclerotome. Here you can see this is the ventral region. It becomes mesenchymal to form the sclerotome. Cells in the upper region of somite form the dermatome. This you can see, this is the upper region. This is a somite as a whole. The cells in the ventral region, they are forming the dermatome. Cells in the upper region, they are forming the sclerotome. The muscle forming areas are formed on the ventrolateral and the dorsomedial lips. Now, a very important differentiation between dorsal and ventral. As I told you earlier, just for the sake of convenience, this is the ventral surface and this is the dorsal surface. It is very important to keep that in mind while development of embryo is being concerned. So, the muscle forming areas are formed at the ventrolateral. This is the ventral part and this is the lateral part. So, the ventrolateral and the dorsomedial. This is the dorsal part, this is the medial part. So, ventrolateral and the dorsomedial lip, it is here where the muscular portion is being formed. Cells from above region migrate ventral to the dermatome to form a dermomyotome. Now, cells from the above region, they migrate ventral to the dermatome. This is the dermatome, they migrate ventral to it. Now, this is what a myotome is. This is how, this is the originator or the precursor of the muscle. Now, cells from the ventromedial region also migrate into adjacent parietal layers of the lateral plate mesoderm. So cells are also not only migrating to form a myotome, they're also migrating to the adjacent parietal layer of the lateral plate mesoderm. They form the infrahyoid. Infra below the hyoid, abdominal wall, the wall of the abdomen, and the limb muscles. So the cells which migrate adjacent to the parietal layer of the lateral plate mesoderm, they form three structures. Those structures are infrahyoid muscles, the abdominal wall, and the limb musculature. So remaining cells in the myotome form muscles of the back, the shoulder girdle, and the intercostal muscles. So again, the cells that did not migrate, what do they form? They also form three separate structures, three separate muscle groups. These muscle groups involve the muscles of the back, the muscles of the shoulder girdle, and the intercostal muscles. What are the intercostal muscles? The intercostal muscles are those muscles which are located in between the ribs. Now, lateral somatic frontier, a very important term. You must always remember that it is the border between the somite and the parietal layer of the lateral plate mesoderm. So what is lateral somatic frontier? It is the border between somite and parietal layer of the lateral plate mesoderm. It is initially very well defined. It forms the mesodermal domains in the embryo. There are two mesodermal domains, the prime axial domain and the abaxial domain. So the prime axial domain comprises of a region around the neural tube. It contains only somite-derived cells. So this is the neural tube. The prime axial domain consists of the region around the neural tube. It consists only of the somite-derived cells. The abaxial domain consists of parietal layer of the lateral plate mesoderm together with the migrated cells. We talked about cells migrating to the lateral plate mesoderm. So the abaxial domain consists of the parietal layer of the lateral plate mesoderm along with the cells that have migrated into it. These cells migrated from the somites. Now, muscle cells that cross the lateral somatic frontier form the abaxial muscle cell precursors. Obviously, all muscle cells originate from these somites, from the myotomal segment of the somites. We talked about cells migrating from the myotomal segment into the parietal layer of the lateral plate mesoderm. So the muscle cells that migrated across the lateral somatic frontier, they form the abaxial muscle cell precursors. They receive differentiation signals from the lateral plate mesoderm. Under its influence, they differentiate into their specific muscle groups. Cells remaining in paraxial mesoderm form prime axial muscle cell precursors. So the cells that remain within the paraxial mesoderm, they form the prime axial muscle cell precursors. Now, innervation of the skeletal muscles. Each myotome receives its innervation from spinal nerve, which is derived from the same segment as the muscle cell. So, the muscle cell originates in the same segment as the spinal nerve, and each spinal nerve individually supplies the muscle. Abaxial muscles, that is the muscles that lie above the axis, that is, they lie at the back, are supplied by the dorsal primary rami. Hypaxial muscles, which are the muscles that lie below the axis, which is the body wall and anterior body wall and the limbs, they are innervated by ventral primary rami. So during development, the myoblasts fuse to form long multinucleated muscle fibers. As you can see in the figure, it is a long fiber and has multiple nuclei in it. Myofibrils appear in the cytoplasm, so they are cytoplasmic elements. Cross striations appear by the end of third month. So this was all about this section. I hope you enjoyed it. I try to keep it as simple as possible. Try to understand and try to memorize these, as this would be very important in the clinical correlates. Thank you. Keep watching sqadia.com.